The first lecture we were going through, the different planes, the coronal, the dorsal, the ventral, the rostral, the caudal. Other terms that I'm going to use very frequently is ipsilateral versus contralateral, and I cannot see anything. Hopefully it'll, there he goes. So the problem is I can't see my pen up here. It's like a 5-second delay. So ipsilateral and contralateral. Ipsilateral means same side. Okay, contralateral means opposite side. So if this projection or this lesion is ipsilateral to something else, it's the same side to it. Okay. The other thing that I need you to know is efferent versus afferent. Okay, and hopefully that'll appear. Here we go. Efferent means away, and afferent means towards. So if I have a cell, or if I'm talking about a projection that is afferent towards, let's say, the frontal lobe. So these are afferent projections. That means these are projections on their way to the frontal lobe. But if I say efferent projections from the frontal lobe, that means they're coming away. They start in the frontal lobe and they're moving away from it. So efferent, E for exit, afferent towards, and then ipsilateral, contralateral. If you weren't already familiar with those terms. Okay, that lecture is done. So we're going to go on to this lecture, and we're having serious problems, so bear with me. So we're going to go into the cerebral cortex. We're going to go from top and then start diving in towards the core of the brain as the lectures go on. So for the cortex, you just want to appreciate that we are not a homogeneous cortex. We have 6 different layers. And what I'm showing you here, and I'm going to write very little, so you guys keep writing because there's the problem I'm having. A Golgi stain is an interesting stain that was discovered kind of by accident. It has this property of probably staining about 5% of the cells it comes in contact with, and when it does stain that cell, it stains the cell in its entirety. So Golgi, hundreds of years ago they discovered this, so it creates these big, beautiful cells. You can see from beginning to end and all their little details. What you don't see are the 95% other of the brain cells that didn't pick it up. So they look very sparse and kind of isolated, but in truth, there's everything in that space is packed. So a Nissl stain, which is this middle column, is a basophilic stain, which means it likes to adhere to its opposite. So it likes acidic properties. So it likes DNA and RNA. So when it binds, it's binding to those DNA and RNA structures, the endoplasmic reticulum, the nucleus, etc., the ribosomes, and so that you tend to see only the core, what's in the most center of the cell body. So we see cells as just spots rather than these beautiful processes. And then now you can see that they'll pick up all of the cells, right? So long as that dye can penetrate in, they will stain it. And then this last column is a Weigert stain. It's a myelin stain. It only picks up that oligodendrocyte, that lipid kind of covering. And so you can see now there is a whole lot of traffic around these neurons in every possible direction. So these are happening simultaneously, but with different stains, we can kind of look at a different perspective. Now, going into the 6 layers, we're going to talk about each layer. I will write, it will come out sloppy, but try to just hear my description and make your notes this way. The molecular layer is a very cell-poor layer. There's mostly dendrites and axons in it. So you can see here when we look at the Golgi, there's really nobody there. The Nissl stain, a couple of little speckles, but in the Weigert stain, we see lots of traffic, lots of axonal myelin. Okay? So that's the thing to know. The molecular layer, very little, no cells, all traffic, axons and dendrites. Okay? That's what you got to remember. This is just a memorization task. External granular layer. These are small, pyramidal, neurons. And they are, I will wait till it appears. These are mostly cortical, meaning that they only transport information from cortex to cortex. It doesn't go anywhere else. So it's just giving sort of local connections and connectivity. Okay? The external pyramidal layer is also similar. These are larger pyramidal neurons. And these are the majority cortico connections. Okay? So these are very similar. The external granular layer just has smaller pyramidal cells, and then the external pyramidal layer has the larger pyramidal cells, and you can kind of see that here, right? These are smaller, these are much larger, okay? Then we go into the internal granular layer. And so these are granule cells, and what's interesting about these cells is their main function is to receive inputs from the thalamus. Okay? Oh, look at that. It crashed. This will really suck if we do the old-fashioned way. Is there markers? Okay. So that's the internal granular layer. We are here. So the internal pyramidal layer is, again, another pyramidal neuron layer. And these are projecting to subcortical structures. So anything that's not cortex, okay? So that would be brainstem, spinal cord, basal ganglia, but what it doesn't do is not the thalamus. Okay? So, pyramidal layers, these are not cortical, these are pyramidal neurons that go to subcortical, which means anything but a cortex, and then things like basal ganglia, the brainstem, etc., but not the thalamus. And the last layer is the polymorphic layer, and these are also pyramidal neurons. So you'll notice that these pyramidal neurons have adjectives. They're the small ones, they're the large ones. The polymorphic gets its name because the pyramidal neurons have all kinds of different shapes. They're fusiform or multiform, they look like spindles, so this is kind of a hodgepodge of shapes, so they get into this last polymorphic category. And so these neurons go to the thalamus. Okay? So, you have, I guess we disappeared it. The granule cell layer is from the thalamus. Okay? The internal pyramidal is all other places but the thalamus, and then this final polymorphic layer is to the thalamus. These other two are cortical, just small and large, and the top one, the molecular, no cells, they're really just traffic, highway space, okay? So this is pure memorization. You can see what the exam test will be. Question would look like, you know, which of these send projections to the thalamus, period. Very simple, okay? All right. Please change slides. All right. Now, just to give you a sense that the 6 different layers that are in the cortex are not the same proportion throughout the cortex, throughout the brain. So we can see here are 6 levels, 1 through 6, and in some areas, so this is a cortical section, they're proportionally larger than in other areas. They're the same layer 3, but they're slightly different in that kind of distribution. And the granular, the agranular, these tend to be more of the motor cortex areas, whereas the granular, these tend to be more of the sensory type of cortical areas. Okay? They're not, it's not 100%, but generally this is the kind of broad general rule. Okay? So when we are looking at cortical neurons, about 80% of all cerebral cortex neurons are pyramidal. Okay? They just come in all kinds of different shapes and sizes. Okay? So we're going to leave the neurons, the cell bodies of the cortex now. We're going to go into those fibers. So we have 2 kinds of fibers in the brain, association fibers and commissural fibers. So we're going to learn the association fibers here. There are a number that you have to commit to memory. The first one is the superior, let it show up, longitudinal fasciculus, also known probably by the more common name, arcuate fasciculus. Okay? So you can see the superior longitudinal fasciculus or arcuate fasciculus here in this dissection. So it has where it gets its name arcuate, which means arch. You see this very large arch that covers and connects frontal lobe, parietal lobe, occipital lobe, temporal lobe. So it is the major highway that the brain can use to get from front to back and cover every possible lobe. In this picture, it's the gold labeled, the yellow labeled pathway is the arcuate. If you hone in on your tablet, you can see a little A in there. So that's for the arcuate, okay? So you have to remember these association pathways are not an A to B situation. It is a general, like our highway interstates, where we have lots of on-ramps and exit ramps along the way. So fibers will jump on and jump off anywhere along this pathway. So it's not about who's in the front and who's getting at the information at the end. It's got a very diverse, lots of bundled fibers. It just happens to travel together in sort of this parallel manner. So that's one that you'll need to know. The other is the superior and inferior. It'll better show up. Inferior, did I show up? Inferior occipital-frontal fasciculi. Okay, hopefully all of that will show up. So this is the superior and inferior occipital-frontal fasciculi. It seems to pick up half of the right. So the superior is this gold one in the back, and the inferior is the orange one in the front. So they sort of sit a little bit like this. One is higher, and one is lower, and one is further towards the middle, and one is further to the lateral side. So you see the yellow is the higher one, the orange is the little lower one, so that's why this gets to be called superior, inferior, occipital-frontal, occipital-frontal. So it's a very parallel, kind of straight front to back pathway. I'll show you in the next slide a better picture of it. Over here, we have the cingulum. We had sort of talked, I know I talked about this with a couple of groups. So the green here is the cingulum. It is inside the cingulate gyrus, which is the first gyrus that sits right above the corpus callosum. So right underneath here is the corpus callosum. And then that first bulge, it runs from the frontal lobe all the way technically into the temporal lobe. But mostly it looks like this kind of front to back. There's a little bit of fiber that goes in the temporal lobe. The cingulum is a really important limbic structure, so we'll come across that a lot, okay? So those are the ones that I need you to memorize, okay? Superior longitudinal fasciculus, people like to use this and sort of trick anatomists. And it's like, where is that? Well, I don't know, but I know where the arcuate fasciculus is. Trust me, I see this happen. So I want you to never, after taking my class, never to be fooled by those folks who want to suggest that these are different. They're the same. Same with the superior and inferior occipital frontal fasciculi, because there's two of them, and then the cingulum, and then this is just another example. This has everybody in it. Now the last one I want to show you is the uncinate. Uncinate fasciculus, which is this red one. And so we talked, I know, to some, we were looking for the uncus. We kept calling it the hook. And I know it doesn't make any sense when it looks like a little bump, like your thumb knuckle, but this is where the hook comes from. The uncus, or the uncinate fasciculus, starts in the frontal lobe and then quickly whips around and goes into the temporal lobe. So it has that sort of, looks like a hook. So it's a quick connection between the front of the brain and this sort of side temporal lobe. So it goes straight in there, and that's where the uncus gets its name from, the uncinate fasciculus. Okay? Any questions about this crazy technology that's not working? You can't read something? Aya.
What did you say the cingulum sits on top of? Which?
The superior occipital frontal. So this gold one back here, you can see if you look real close on your tablet, it says SO. So the superior occipital is more superior than the inferior occipital frontal. So they kind of sit like this. And then medial laterally, the superior occipital is more medial. So let me show you the next picture. So here, this is just the superior inferior occipital. So this is them separated. Here's the superior occipital frontal, and this is the inferior occipital frontal. When put together, ooh, my pointy, you can see them in this dissection. Okay, so back here, this is the superior, and then down here, sweeps under is the inferior occipital frontal. And they're basically big connections from the frontal lobe to the occipital lobe. Just they're parallel, just a little offset, and a little higher and lower than each other. Okay, so not quite train tracks. Okay? Any other questions? Good question. Okay. Here is a video of the white matter. So we're going to, oh, it not play. It may not play. All right. Well, we'll try to play it another day. Everything about this computer is like super slow. We sat here all day. Anyway, the whole point of that video was to show that the brain is mostly white matter tracks. We think that we talk about the cells and the cerebral cortex, and we think it's all that kind of mass, but it turns out most of the mass of your brain are the trafficking pathways. Okay, so that video showed it in sort of a nice way. Okay. Those are association cortices. Association fibers connect a place from one to another, but within a single hemisphere. Did I fail to mention that? They're all in the same hemisphere. So when we talk about the arcuate, the superior inferior occipital frontal, uncinate, they're all confined to their specific hemisphere. Then the other hemisphere has its own version of that same fiber pathway. Now contrast this with commissural fiber pathways. Commissural fiber pathways, there should be a C, are pathways that cross and connect one hemisphere to the other hemisphere, okay? So we have three commissural pathways that are important to us. The anterior, the posterior commissure. The anterior commissure connects, it's up in the front, okay? So let's assume this is front, it's not a terribly accurate brain. It will be up in the front. It connects the two temporal lobes together, and it will connect the limbic structures like the amygdala together. And who uses it? Obviously the two temporal lobes use it to kind of talk to each other, but olfactory information. So when olfactory information comes through that olfactory bulb, it jumps onto the anterior commissure, and then that's how we get olfactory information to limbic areas like hippocampus and amygdala right away. They've got a very fast track path to get there. The posterior commissure, as you would imagine, is out in the back. It sits in the midbrain, so just in front of the cerebral aqueduct. And it's an important area for cranial nerve 3. So it allows our eyes to operate in a very parallel manner, so we don't have gecko googly eyes that get to go in every direction independently. Our eyes are always tied together. When one moves, the other moves as well, and that's because of the posterior commissure. Now, the big one is the corpus callosum, which all of you probably know, and that is this guy in the middle. It is the one that allows the two hemispheres to connect with each other. It turns out that these two hemispheres are somewhat independent. Even though they're structurally symmetrical, they're kind of built in a twin-like manner, there are differences between the two hemispheres in terms of what type of function that they control. And so in order for one side to know what the other one is doing, they use the corpus callosum. Now, what's interesting in how we know that is through situations where we have had to manipulate that corpus callosum and do a callosotomy, meaning we cut the corpus callosum deliberately, and these are usually the extreme last-ditch efforts people will use to control epilepsy. So if you imagine epilepsy as a prairie fire, once you start the prairie fire, the electrical storm, the goal is to shut it down as fast as possible so that we don't sweep the electrical storm across large areas of the brain. So if we have a sweeping electrical storm, one way to stop it from getting to the other side is to cut it here. So that is the extreme measure some epileptics are given. And it is a very effective way of controlling epilepsy, because once you start somewhere and you can't get across, the electrical storm ends. So, but what's interesting in these callosotomy patients is that there were things that we learned from them after, we did that for practical purposes, but then we went and tested them about, hey, how's your brain different? Most of the time, a corpus callosotomy, you can't tell in an individual, they function just fine, because most of all of our interactive points in our brain are bilateral. We have two eyes, two ears, two sets of hands, and two sets of sides. So most of the time you can't tell because both sides of the brain get that information. But if we experimentally prevent that from happening, we get some very interesting results. And one experiment was to prevent someone from seeing something in a different visual field. So, for example, things that are in the left side of your visual field. So if you took a middle cut down your perspective, everything on the left side is really going to your right brain, and everything in the right side, right visual field, is heading to your left brain. And so the left visual cortex and the right visual cortex get different parts of your field of view. So if you were to hide and put a word in the left visual field but not the right, that word is transmitted to the right side of this brain. But remember, I haven't told you, here's a little extra fact that we'll get to in a few minutes. Language is primarily located on the left side of the brain, not the right. So now, because this is a word, I need to get it over to the language center to be interpreted, to be identified, to even be detected. So in a callosotomy patient, when they're shown a word, they're like, I didn't see anything. Really? You didn't see anything? It flashed in front of you, it stayed there, it wasn't subliminal, it was on there, it's sitting there now. I don't see it. The word is sent over to this brain, right brain, but because of the cut, it cannot transmit it over to the language area. So there is a word agnosia. They don't see it. What's interesting is they can then take a picture, you know, a cartoon or something, show it on this side, and again, the picture is transmitted to the right brain, but the person still can't identify it. They can't even tell you that they have seen anything. But if you ask them and give them a tray of objects, and one of them is the object is in this picture, they can pick it out. They'll just pull on it, and it's like that one. But they don't know why, because they can't tell you, because they can't get it over to the language area. So they'll always tell you, I don't know what it is, and I don't know why I chose it, but I've chosen it is because the information still made it over to the right side, but because of the break, it cannot go to the language area where it can be verbalized. And if it can't get to the language area, not only can't you verbalize it, but you can't acknowledge it. Crazy, huh? Any questions? Okay. So, just to illustrate the importance of the corpus callosum, the fact that our two hemispheres basically operate independently, but they're very cooperative because they can share that information across that bridge. Okay? So, now we begin to get into the idea that my two hemispheres are independent, and I told you before that they are functionally specialized. So for example, language is mostly found on the left side of your brain. That's 90% of those of you who are right-handers, 90% probability that language is on the left side of your brain. For you left-handers, it's still Felix, your majority is still likely to be language on the left side, but little less chance. About 70 to 80% of left-handers will have language on the left side. So it really doesn't matter which side it's on, it just happens to be mostly left side. Wherever language is, we call that your dominant brain. Okay, so whether you're a left or a right language person, that's your dominant brain. Okay? I can tell I'm going to run late already because I am just having on. Okay, here's my quick review. We did 6 cortical layers, 5 association fibers, and 3 commissural fibers, and a partridge in a pear tree. That's all. You just make some note cards, just write them down, memorize them, boom, you got that, okay? Nothing more complicated. Okay, so now we're going to get into delineation of the cerebral cortex itself. And so we can divide our brain into primary cortices. Show up. There we go. In the primary cortices, we have sensory, visual, auditory, and motor. Oops. Okay. Outside of a primary cortex, we then have association cortex, and limbic. Those are our 3 big categories. Now, if you are a primary cortex, you have a very delineated special place for these 4 functions. However, association and limbic are a little more broad, and what's counted as it. Association cortex tend to be higher order cortices, meaning they do things that make us more human. This is where we find art and music and sort of imagination and very complex sensory motor coordinated functions. Limbic cortices are those that are involved in emotional processing, either understanding the world and the emotional information that's being sent to me, or creating my own emotional message and sending it out. So the motor and the sensory side of emotional processing. So this is an example of the motor and the sensory primary cortices. You can see here they're very narrow. It's just a strip. So in the red we have the motor gyrus and the sensory in blue. So the divider, of course, is the central sulcus, and the precentral gyrus here is the motor. Postcentral gyrus is blue. Has anyone never heard this? Okay, good. So this is your homunculus. I assume everybody knows. Anybody not know what a homunculus is? Don't be shy. Okay. So a homunculus is it literally means little man. And so this the little man that lives in either the motor or sensory cortex. So this is a representation. This is the motor homunculus, and this is the sensory homunculus. You can see your whole body is represented on this strip of brain. And that location tells us which part of the cortex is associated with which part of your body. So the motor and sensory cortex are highly, we call, somatotopic organization. There we go. Somatotopic meaning what is next to my body. So let's say my forearm is next to my elbow, which is next to my bicep, and in that order. I will find that same organization on my homunculus. Okay? So you can see here we have, it's not quite distributed head to toe. There are kind of some rearrangements, but generally inside this longitudinal fissure, like inside the divider, is most of our lower extremities. Okay? Outside on this lateral convex will be our upper extremities and our head and face. That's generally the layout. And then what makes the homunculus look so distorted is the bigger the body part, the more sensitivity or more capability you have. So for example, in motor, your hand is very big. You have a lot of dexterity and precision and control in your hand, and you have literally none in your bicep because it's either on or off. It's either contracted or not, but there's nothing else it can do that's crazy talented, okay? So we have a lot of emphasis in our face. We have a lot of muscles and a lot of ability to detect. People can detect an eyelash sitting on your cheek, right? So that's a lot of sensitivity. So that's why these are very large, right? All right. So we'll find that there is that same organization in our primary cortices that you see here. So obviously sensory and motor has a somatotopic organization. Your visual system has a retinotopic organization, and the auditory has a tonotopic organization. So for example, the retina that's in the retinal field that's in the back of my eye, whatever arrangement that my visual experience is at the retina, it will transfer that same shape and arrangement in my thalamus and then all the way to my visual cortex. It will keep that same proportion and arrangement. Same with auditory. The tones from low frequency to high frequency are represented all the way through the different stopover points. And they don't get mixed up, they don't get reshuffled, they stay in that tonotopic arrangement. So that's a really kind of nifty feature of our CNMs. Okay, so let's start with the sensory cortex. I don't remember. Okay, so our primary sensory cortex, we will get into this really detailed, so I'll just give you a preview right here. We have 3 neurons that get information from our body, our sensory mechanoreceptors, to our brain. So you'll get to know them in sort of a way that you can predict exactly their location, but a sensory mechanoreceptor then sends its information to the spinal cord, it goes to the brainstem. The thalamus is the first place it stops off, and then it goes to the somatosensory cortex. This is Brodmann's area. You will learn, must learn, Brodmann's area, 3, 1, 2, okay? So these are part of the few Brodmann's areas you're gonna have to pick up. So basically, it's that simple. The thalamus is an important kind of way station for sensory information. It takes almost all sensory information has to go through the thalamus. Now, if I stimulate Brodmann's area 312, what would happen? Well, you can either get sensations of tingling, depending on where in the homunculus you're stimulating. So you might get tingling, or you might get sense of numbness. So it's not always one or the other, sometimes it's both. It starts out tingling and then it gets numb. So it's just an indication of how complex even the 312 is in terms of detecting sensory information. Now, if I lesion Brodmann's area 312, we want to really know what this means because we're going to have patients at the latter half of this class that we're going to have to figure out what's going on. If I lesion or damage Brodmann's area 312, I will lose quite a lot of detailed sensory processing. So for example, I might lose proprioceptive or stereognosia, meaning I won't be able to detect something by touch. So this is a simple game we play with students. We put your hand in a bag and you have to feel your way. Well, it's a paper clip, I feel a pencil. And so you do this only by the sense of touch. That is what you're going to lose in here. But interestingly enough, you will never lose 100% of your sense of touch. It'll feel like, if you remember going to the dentist, you get a shot of Novocaine, you really don't feel what's going on, but if your friend is poking you, you do know that your friend is poking you. You just don't feel it in great detail, but you know, okay, you're poking me now. Okay, now you're doing it again, right? There's a broad, very sort of low detail sensation that remains. So when you lesion 312, you never become 100% without sensation. However, somatosensation is not just fine touch, it is also proprioception and vibration. Proprioception requires stretch receptors and skin stretch to tell me that my limbs are moving or my limb is high up. And even if my arm is raised and my eyes are closed, I know it's up there because of the stretch that's happening in different joints. That will be lost. That, when you damage 312, that can be obliterated 100%. So that becomes very challenging to an individual who can't feel their body. They have a lot of difficulty getting across the room just walking. If you cannot feel where your body is, you have to literally look at everything. You have to see where your foot is, make sure it reached it, then decide to bend something just enough to get to the next step. So it's challenging without that sense of proprioception. Okay? Any questions? Okay. The primary visual cortex. This is Brodmann's area 17. Okay? The visual pathway starts from retina to thalamus to visual cortex. Hopefully that will show up. Not really. Visual cortex. Okay. So light enters your lens through your pupil, hits the retina. That's our first cell that's being impacted. That information is sent to the thalamus, a special part of the thalamus called the lateral geniculate nucleus. And then that sends it through two different routes to your visual cortex. So it's the retinal, geniculate, striate, or cortical pathway. That's what we call the visual pathway. And so 17, Brodmann's area 17 is the first cortical area that gets activated by any visual input. Okay, so that's our primary visual cortex. After that, that visual information starts being sent forward again. So we then have 18 and 19. Those are the association cortex, not primary. When we get to the association cortex, we start adding on information, making that complex. We add color, we add form, we add depth, we add motion. So, but in the 17, it's just brutal, on, off. It's there, it's not there, okay? Questions? Any uncertainties? I talk fast, it's okay. No? Okay. Now, if I damage 17, I will get, if I completely take it out, you'll get cortical blindness. Okay? That means that you have no, you have no conscious awareness of any visual input. Okay? So without 17, all these other visual areas that are coming downstream will get nothing. There's no way to bypass that. Or is there? But that's a different class. Okay? But right now, this will be leading us to cortical blindness. Okay. Let's do our primary auditory cortex. Our primary auditory cortex obviously starts with the cochlear cells in your cochlea, the hair cells in the cochlea in your ear. I will learn their pathway near the end of the class, but they end up here at 41 and 42. So technically, this is in your temporal lobe. Okay? So remember, here's 312. We know that's in our parietal lobe. Okay? So 41 and 42. So Brodmann's area 41 is our primary auditory cortex. And then it sends its information to 42, which is an auditory association cortex. So we add more stuff to it, and it can process more complex. And what's nice is right next to 42, there is a neighbor, 22. Okay, so you can see that here. 41, 42, and here's the neighbor, 22. 22 is Wernicke's. We're going to talk about that in a second, but Wernicke's is a language comprehension. So one of the primary ways we comprehend language is through audition, so it makes all kinds of sense that they're right next door neighbors. But Wernicke's is not an auditory processing. What it does is it's, I can use that information for my benefit, okay? So 41, 42. I've already mentioned that it's tonotopically mapped. So we have mentioned that the retina sends its first stopover, synaptic stopover to the lateral geniculate nucleus. In a parallel form, the cochlear hair cells send their first, the second stopover to the medial geniculate nucleus. So these two little parts of the thalamus have a sort of a parallel organization, one for vision, one for auditory. So the medial geniculate nucleus then goes to Brodmann's 41 to 42 and then onward. If you damage the 41, let's say we obliterate it with a stroke or some other brain damage, you do not get an appreciable hearing loss. That's an interesting fact. Unlike the other senses and the other primary cortex, you don't, because auditory information from your two ears join together very, very early on. So most of the pathway that you get from the cochlea to the cortex is already carrying two ears bilaterally. So once we damage one, the other 41 on the other side already has information from both ears. So losing a 41 doesn't get you a very big deficit. The two things you do lose is the ability to tell foreground from background. So this is when you're in a busy, loud coffee shop and you're having a conversation with someone, you've got to kind of drown out the background noise from the foreground conversation. That becomes more difficult because you don't have that distinction. And the other is echolocation or sound, auditory location. When someone yells, "Hey you," you generally turn towards the sound, the shortest distance to the "Hey you." Without your two areas of 41, you're a little less, you don't really know where the sound is coming from. So locating sound and then drowning out the background noise. Okay. All right. Now, we're going to do the primary motor cortex next. The primary motor cortex is Brodmann's area 4. Remember, it's the pre-central gyrus. Here's our central sulcus between the red and the blue. The blue is our somatosensory, 312. Our red here is Brodmann's area 4. We know that we have a motor homunculus represented, so the whole body is represented in that thin strip. Okay, so that's our primary cortex. We also have a motor association cortex. These are Brodmann's area 6, and we divide 6 into the lateral part of 6, which is called the premotor cortex. And then the medial, and this is called the supplementary. Okay. So you can see the red is Brodmann's area 4, and then right in front of it is Brodmann's area 6. This purple and this yellow is all 6. But the lateral, which is the orange area, the yellow area, is the premotor cortex. And then the purple area, not only is it at the very medial at the top, but then it dives into the big divide. So here's the rest of 6. This is the supplementary motor cortex. Okay. So what happens when we stimulate 4? We generally get a tiny muscle movement. That's about it. Nothing very large, a single muscle, a single joint, very small movement. And then it's often followed by some kind of paralysis. Like you don't continually get a lot of motion or tremor when you stimulate this area. When you get to Brodmann's area 6, the premotor, you also, when you stimulate that, you also get muscle movement, but these are larger muscles. Maybe you get a hip flexor or a shoulder movement, so these are larger limb motions, and maybe take multiple muscles to pull off, right? So just to lift the shoulder up might take 1 or 2 extra muscles than a single movement of a digit. Then the medial or the supplementary Brodmann area 6 is similar. It's more muscles, and then these muscles tend to be parallel, symmetrical. So then these tend to be postural muscles. Things in your torso don't ever work alone. When you bend your ab or you pull on your back, it's always in a paired situation. It's not possible for you to contract one ab muscle without the other ab muscle. So supplementary motor cortex tends to govern anything that's in paired motion, and so this is more postural control is in here. So if you damage Brodmann's area 4, let's say with a stroke, you will get paralysis. And where you get paralysis is dictated by where in the homunculus you are. If you get damage to premotor and supplementary motor, you don't tend to get paralysis, but you will get weakness, so paresis. So anytime you damage this level of the upper motor neuron pathway, the first neuron that's sending motor information, you'll have a kind of a sequence of symptoms. It tends to be flaccid paralysis, which means flaccid paralysis. Flaccid meaning no tone, kind of like a rag doll. Okay, so you'll have this flaccid, no tone control, and of course you're not able. You have the paralysis. Then with time, weeks, months, the flaccid tone will turn around and become really hypertonic. So then you'll become really stiff, and that stiffness plus the weakness gets you something called spasticity. So spasticity, individuals who have maybe damage to 6 and not 4 might have some weakness where they don't have good control, and because of the tone, they don't have fluid movement of muscles, so they're more lurching. They're using muscles in like a big bulk unit and sort of picking things up rather than using them in a nice sequence. Owen.
Does it affect people's diaphragm?
It does affect people's diaphragm, but you have to really catch it at the neural activation. So that is C5, C6. So you have to damage those motor neurons. The diaphragm itself isn't easy to kind of damage through the premotor cortex or the primary motor cortex. Any other? It's a good question. Anyone else? But we will talk about when your diaphragm is at risk. Okay. let me make sure I'm picking up everything I'm supposed to tell you. Oh, remember in both the somatosensory and the motor primary cortex, whatever side it is on the brain, it's affecting the opposite side of the body. I don't want to assume you know that, but that's probably something you've been told earlier. There's always a contralateral control. So sensory information from the left side of my body is going to my right BA312, and then my right Brodmann's area 4 is controlling the left side, the muscles on that side. Okay. In addition to spasticity, the increased muscle tone due to damage with either 4 or 6, another thing that you'll see is an increase in all your deep tendon reflexes. So that's a hallmark collection of symptoms that you're going to want to see every time you suspect damage to the brain. I want to see spasticity, which is increased tone, decreased weakness, or increased weakness, decreased strength, and then an increase in reflexes, tendon reflexes, bicep, patellar, Achilles. All of those should be exaggerated, really fast, kind of abnormal that way. So hyperreflexiveness, the increased tone, and the lack of muscle strength leads to that total spasticity presentation. Okay? Okay. All right. Okay, I'm a little over, so we'll pick this up. Oh, here's my good point to end. Our primary cortices we've covered are sensory, visual, auditory, and motor. We'll pick up the rest of this lecture next Friday. This material, everything that we've covered so far, is fair game for Wednesday's oral quiz.